Okay, so this talk is about particle tracking, and I will first of all establish the importance of particle tracking in our, I say, in our society and so on. And can you get the next slide? Okay, so particle tracking, of course, was one of the earliest things that the men and women did. And so those are two stamps from Aristoteles and Ptolemy thing. And you can see the complexity that they end up by putting the earth in the middle. And of course, you know, can you get the next slide? And so when you put the sun in the middle, everything becomes much easier to describe. And you can see in those two things what happens with the eyes, what happens with the sun is in the middle, and the earth is in orbiting around the sun. So next slide. Okay, and I want also to have Luis Alvarez, which has used particle tracking to look at the very, very small world of the elementary particles. And so you can see that going from the seasons to elementary particle tracking has always been very, very important. And so we added a little bit to that. And so can I have the next slide? Okay, we added a little bit of that by describing essentially how you do particle track. So you can do, this is a slide of the 60s, optical trapping. You can do very, very, very fast, very high speed, but very small range. Orbital tracking, you can do essentially the same speed that you can do optical tracking, but at a very large range. And then you have other techniques that I will not talk about. So next slide. Okay, orbital tracking has been available for quite a time, and probably one of the very first things was to using a video camera, and this is tracking of colloidal gold particle 40 nanometer in PTK1 cells. And you can see the tracks here, very well defined. And there are many other papers. One paper describes essentially the observation of the particle and the tracking of the particle. And you can go to the next slide. And the next slide describes the fundamental equation for particle tracking that says that the error that you get in particle tracking depends on the error of your microscope, depends on the inversion of the number of particles, and then on other factors. And those are been known for quite a time. And those equations are true, and there is nothing else you can do. But let us see what is the duration of track. So duration of track. So if you use cameras, it's very small because you can observe only for a certain period of time. And then if the particle goes up or down from the field of view, you miss it. And then you can see that the duration of frames is at the most of the order of hundreds. You can do 100 frames, and you cannot do more than that. And this is very important because you can observe a particle diffusing for much, much longer time with a technique I will describe in a minute. So can you get the next slide? Okay, so now the issue is how to do 3D particle tracking. And people thought about that, and there is a relationship between the focus, the kind of focus that you can get in the Z-axis, and clearly when you are at the focus, you get very small interference pattern. And when you are out of the focus, you get a lot of interference pattern. And you can see that you can get at the most 3 micron out of the focus and using this technique. This technique works, but works only for a very small amount of. Can I get the next slide? Okay, so tracking a particle can be much faster. For example, this is the particle in which you have a particle which is trapped, so it's trapped in an optical trap, and then you have a quadrant a photodiode, and the quadrant photodiode is the particle move to left to the right, it will change, and then you can do very, very fast. And that was described in 1999, and you can see that using this technique, you can track a particle, a movement of the particle on the order of 50 kilohertz, which is very, very fast. Next slide. Okay. The other possibility is to track the particle. So as the particle moves from one point to another, you change the z-axis, and then you can obtain track the particle. And this is a paper we published with Peter So and Tim Regan in 2006. And of course, you need to know where the particle is going. So you can go 1 frame up and 1 frame down, and essentially you can try to track the particle, but still, you lose a lot of time because having a frame up and frame down, it takes about 3 to 6 hertz response time. So this will not work for a normal particle. So next slide. Okay, now let us go to the principle we are using. So this principle is really, so in red here we have the volume of illumination along that line. And then you can see that the volume of illumination has so if you change the volume of illumination in this region, the derivative is 0 because there is no change. And the maximum of the derivative is in this part. So you would like to have a particle which is exactly in this position, because that will give you the maximum change in the derivative. And if you are able to do that, then you can maintain the particle in the derivative. So let us see how you can do that. So can I get the next slide? Okay, so suppose this is the microscope volume of illumination, and you are able to rotate the volume of illumination in an orbit. So when a particle is, for example, in the red point, you will have, as you move the orbit, that the particle will have a maximum intensity at this position, this phase. And if the particle has a different phase, for example, close to the orbit, you will get a larger signal. And if the particle is in a different position, the phase will change and the amplitude will be whatever it is. So that represents essentially the origin of this kind of technique. And although people in Germany realize that you can do that, they never developed the method in the same way that we developed. Okay, so next slide. So now let us see in a moment what's happened. So what you measure as the position along the orbit changes, so the position along the orbit changes, you have a maximum in AC of the signal, and the maximum, so the DC will be the average of the orbit multiplied by 0.5, 1/2, and AC will be the maximum of the AC to respect to the DC, and that is what is called the modulation of the signal. So you have an AC which is given by the position of the particle, and a modulation which is also given by the position of the particle, but contains a DC. And then you have an angle, phi, which tells you where the particle is to respect to the orbit. And that is the formula we use with Kispetikova was a postdoc in my lab at that time. And we published a paper in which we say that the total intensity is given by 1/2 the average value of F, and then cosine and sine of theta, which depends on the angle theta, and then contains higher orders that we don't consider in this presentation. Next slide. So when we do orbital tracking, so we take an orbit, so particle is in an orbit, and then we will see here, let us see, can I get the next slide? Okay, so the next slide shows how the modulation changes and how the intensity changes. Okay, so if we are exactly in the center, there is no change. As we get out of the center, the modulation starts to increase, and we'll get a maximum when we have this value of the well, it depends on the orbit and so on. So if we got the modulation and the phase, we can tell exactly where the particle is. And notice that the modulation cannot go higher than a given value. So this is important because if we get that value of the modulation, then we will get, we know where we are. So we know the distance, which is given by the modulation, and we know the angle, which is given by the phase. So if we measure the phase of the orbit as we orbit around the particle and the modulation, we can tell everything about the position of the particle. And so this is the method. So next slide. So the method here is now the data. So the method, for example, the modulation, you see, has a maximum when we are out, then it has a minimum when we get the particle, and then it has a maximum when. So it has that shape. So wherever you are, you can go back to the origin. And the radius of the orbit is given by the phase, which can be measured too. So next slide. Okay, so let us see how we can do 3D particle tracking. So 3D particle tracking. So if we are in an orbit which is exactly at the center of the Poisson function, and the particle is in this position, we will have that this orbit, if we move the orbit up or down, so we move the orbit up or down, we have that the position of the particle would be farther now. And so essentially, by changing the focus of the microscope in a given way, we can change the modulation if we go one orbit up and one orbit down. So can I get the next slide? So this method of using a piezo is relatively slow. So a piezo has a response of the order of 150 microseconds. So we decide to use electrical tunable lens, so in which the focus is changed by changing the field, electric field that is applied to the cell. And in that case, we can change the orbit up and the orbit down in the order of about a kilohertz, so which is very convenient. And so the experimental setup is the following. So we have a specimen, which is whatever, we have an objective, we have the Z position, and we go in orbit up and orbit down, and we send the data to the computer, and the computer will decide if the orbit up and the orbit down are different in terms of modulation, and if they are not different, then we are in the center of the particle. If they are different, we will position the orbit in such a way to be the minimum change. So that is the way the computer works. And now let us see how we start. So we start in one position, whatever it is, and so the initial position of the scanner is given to the computer. Then we have the intensity profile that is done by going around. And the intensity profile gives us, after Fourier transform, the phase and the modulation of the system. So if the modulation is too high, it will move in that direction. If the modulation is 0, of course, will not move. That means that the particle is exactly at the center. So let us, can I get the, okay. So let us see how it works. So those are the screen that we have, and we have essentially two curves here. One which says the orbit intensity. So the orbit intensity has to be constant, because that means that we are in the center. And then we have a modulation along the z, so when we move the z, and that also has to be constant. And when the two things are constant, we are on top of the particle. And if we are on top of the particle, as the particle moving the z is shown here, we can track the particle wherever it goes. Okay, next slide. Okay, so essentially, this is the way it works. So we have a particle which is here in B, in A and B. And then we have, for example, a lifetime of everything that can change as a function of position of the particle. And then we can obtain all the parameters like the polarization, the spectrum, and the shape and position. So this is very important, because this is the only method that can give the shape and the position of the particle. For example, if the particle is round or the particle is an ellipsoid, you will get different values of the shape of the particle. So let us go to the next slide. So let us see how the data are acquired. So suppose we have 1, 2, 3 particles here, and then we orbit here. And the orbit will give essentially the same modulation for 1, 2, 3 particles. And the same modulation, remember that we are performing an orbit, and in every point in the orbit, we measure the intensity. So we measure here, we measure here, we measure here, and so on. And this is the chymogram of the pattern. And then we will see some position are very low. So we are in this position, and so on. So, and this is done in 131 microseconds per orbit. So only that part is, this is in seconds, the part of the orbit. So next slide. Okay, so the next slide, from the position of the orbit, we go obtain the DC, and then the modulation and the phase. So we know exactly where the particle is to respect to the orbit. This is very important. And remember that we measure the position of the orbit in many, many points. The maximum the computer can do is 2048. So we can measure in 248 points. And then we can have 248 points in the intensity, and then to calculate the phase and the modulation. Next slide, please. So clearly, as we do the orbit, for example, in that part, there is a change in the fluorescence intensity. So there is a limitation. Photobleaching is a general problem in fluorescence imaging, and always we will have some sort of photobleaching in the system. The other system is that if we use a piezo and we want to move up and down, and we have an immersion oil, as we move up and down, the oil, the oil will stick to the glass and will move the glass up and down. And so that doesn't work very well. But if we use a electrical tunable lens, in which we don't move anything, then we can move X, Y, and Z, and move. So the oil will not make any difference, and that is very important for the So, so let us see what is the problem that we can have in this method. So we have, for example, we have fluorophores, which are 100 nanometers of particles. Concentrations approximately ranging a discriminate individual particle. If the concentration of the sample is larger, this is not possible. So if we have many particles in the field of view, we will have to choose one of the particles. Strategy around this problem may involve photo bleaching section of the sample of blocking photoactivation. And we did that. And then you have 3 particles, but you choose only one to track. For example, you decide which one you want to use. Can I have the next slide? Okay, so let us do an application, because this is very important for the question I asked about the mean flux and the other thing. So remember that we perform an orbit, and this performing an orbit, so this is, for example, a nuclear pore complex, and that will be the cytoplasmic part, and that will be the nucleus part, and we perform an orbit. For example, this is a particle, a nuclear pore complex, and we perform an orbit which is slightly larger than the nuclear pore complex, in such a way that when we are down, we will observe what happens in this side. When we are up, we will observe what happens in the other side. And this is very important because at the same time we do the scanning, we can do lifetime measurement or fluctuation experiment. And this is very important because it depends on the time it takes a particle to go through the pore complex. We will have different results. Can I have the next slide? Okay, so so this is how the nucleus will appear. So each one of those points is a nuclear pore complex, and then we choose one particular point in the nucleus that will be, for example, this particle, and then we have another particle. And the reason to have 2 particles, 1 and 2, is very important because if the entire cells will move, that will move, and then, but the other one will move too in synchrons. And so we have a system that essentially moves the synchronization of the movement of 2 particles, one and the other one. Can I have the next slide? So, for example, we have here 2 particles, and those are organelles among the microtubules. And you can see that one particle can go in one region in the other. And we study those things, and we see that, for example, one particle can have a very complicated orbit, but still we can separate one from the 2 because the track in one part of the orbit, and then the other part of the orbit, when it's fixed in one position, changes completely. So the distance in time changes very dramatically. So let's go to the next slide. So now we go to some different thing that was done by Laura Estrada when she was in my lab, and Chris Hellinger. And we have gold nanoparticle. This is a gold nanoparticle, which you can see here. They seems to move along the microtubules, and they make a trace like this one. So we stop we look for a certain amount of time, it will make a trace like that. So next slide. So the question is, if we are in a microtubule here, and we can see the microtubule with a technique in which we make the orbit farther and closer. So we modulate the position of the orbit, and when it's closer, of course, we have the much larger intensity when it's farther. So we can tell the distance of the object, and the object can be complicated, can be not circular, but can have other shape, by this simple technique that says oscillating the orbit, we can tell where the object is, and that can be done for any shape of the object. So can I have the next slide? Okay, so let us track now some position. So this is a nucleus. The nucleus is expressing MS2 GFP, which binds to RNA, and then there is a lag repressor, M-cherry, which is here. And and we have many repeats of the same gene. So what we have is a lag operator, which has a certain number of a repeat of the lag operator, and then polymerase 3, which will bind to the lag operator and will report the production of RNA, which is measured in different color by MS2 AGF. Okay, next slide. Okay, so now this is a system in which we have many genes, and the genes produce as you can see, a green, produce like petals around the nuclei, and these petals are relatively stable. So now we can measure one petal, and one petal in the same system, and in this system, the RNA is produced, and so we see the green fluorescence coming out from one of the petals. And this is very important because now we can see, for example, this is one site, and we can see, for example, that orbit at a different position. And then for each orbit, we can measure the count or whatever fluctuation of the count. So we can figure it out, for example, that has a maximum here and then goes down to a minimum. And then we can measure the expression. Can I have the next slide? The expression of one particular gene by measuring the autocorrelation curve of these traits. And so, for example, we have large counts, then we have a minimum, then we have a maximum, and then we can filter that curve, and we have an average correlation to it. And this average function of correlation gives us the average time the correlation will have. So the point of 1/2 is about 10%. Okay, so we go and we observe many points of, so many petals simultaneously of the same particle, and then we can observe how long a petal exists. So the kinetics is extracted from the ACF curve. So can we have the next slide? Okay, so and those are now many different angles, many different phasor angle, and then we can measure for many different petals. So there are on the action of the same gene, but now we can observe 1, 2, 3, 4, 5, 6, 7, 8 petals, and to see if the time is different from the same for the polymerase act in a different way for the different petals. Okay, can I have this next slide? Elongation rate is heterogeneous along different cells. So those are different cells, A, B, C, D. And you can see that some points are faster, some points are slower, like this one. And then we can see that the mean, what is the mean value, and then we can build an histogram of all the cell and figure it out how long it takes a polymerase to produce the gene MSG. Okay, so can I have the next slide? Okay. On the same petal, for example, we can measure also how long it takes, for example, from here to here. And we can add factors that affect the polymerization, and then those factors are clearly seen the action of the factor, and say we have an elongation of the MS2, and then the stop when we add the factor. And so here we have the petals. So this is the gene, and it extend a little bit where the polymerase is acting. And then we have 1, 2, 3, 4, 5, 6, 7 gene. And then we can compare one gene with another. So I have the next slide. Okay, so the local mobility of the gene, so the MSD of the gene, the average of all the gene, this is the yellow is the average, and this is the maximum, the minimum, will change depending on the gene, but all the gene more or less have the same kind of behavior. And this is very important because this is the first time that it was shown that although genes can have different time to express, actually, in this case, the genes are very close one to the other, so it does not depend on the availability of molecules. Okay, can I have the next slide? So correlation, so then the other thing that we can do is to see if one petal, one particular gene will correlate with the other or not. So if they are independent or not. And so one moves to the left, the other moves to the right during the gene expression. And so if we do the correlation, the cross correlation among all of them, we get no cross correlation. So they are not cross correlated one with the other. Okay, and that shows. Okay, can you go back? Okay, so that shows the different genes. So some goes up and some goes down. So independently, and then all of them takes a certain amount of time to get expressed, but this is normal, okay? And if you go to the next slide, so the gene in one part, the gene in the other part are essentially elongated with some independent rate, okay? And now the question is, when the MS2 is done, where it moves? So, for example, this MS2 can move in that direction, the other in the other direction, the other in the other direction. And in order to do that, we do a cross correlation between the color of the gene here and the orbit which is passing through. Can I have the next slide? Okay, so the conclusion of this part is that by observing different points in the orbit, we can obtain the rate of expression of every gene. The rate of expression of every gene changes between different petals, but the point in which can I have the next slide? Saying, okay, so SPT for images is well known, but it's also does not work if you want to have very fast and the dynamics of each individual thing. And if we do this lock-in single particle tracking, then we have very high 3D and time resolution, and other information from recorded orbit, for example, lifetime, and the correlation function is selected, and the temporal, of course, is very high resolution because every orbit we can measure the same particle, and so we can come back to the same particle and figure it out if the same particle have a different time than the other. So I think this is the last stuff. Okay, we will stop here for the moment.